that is linkage groups. All genes present on a single chromosome called linkage group. All genes which are present on a single chromosome called one linkage group. Number of linkage groups that is equal to haploid number of chromosomes. Number of linkage groups is equal to haploid number of chromosomes. So, number of linkage groups in garden P 7, maize 10. In garden P, diploid number of chromosomes that is the 14, haploid number is the 7. So, number of linkage groups that is equal to haploid number of chromosomes. So, haploid number of chromosomes garden P that is the 7. In case of maize that is the 10. And in case of the E. coli, only one chromosome. So, number of linkage groups in E. coli, Escherichia coli, only one. In case of man, there is a 24. In case of woman, 23. Then what the difference in between the man and the woman? In case of the man, X and Y chromosome, we must consider these two chromosomes also. 22 autosomes, 2 sex chromosomes, X and Y. So, number of linkage groups in man 24. But in case of the woman, autosomes 22 and sex chromosomes both are X chromosomes. So, consider 1. So, 22 plus 1 that is a 23. Significance of linkage. Significance of linkage. When genes are linked together, they are not involved in recombination. That means they are not separated. They pass to the offspring without any change. So, same characters that are passed into the offspring. So, offspring, they appears parental combinations only. Non-parental combinations that are negligible. If the linkage is weakened, recombination that occurs. If the linkage is weakened, recombination occurs. At that time only, we observe non-parental combinations. When non-parental combination is more, linkage strength is less. So, it was first observed by T.H. Morgan in Drosophila. He observed that when linked genes are present, parental combinations more, non-parental combinations that are less. So, to obtain same parental characters, linked genes are essential. So, linkage that maintain genetic constituency, genetic equality in offspring. So, without any change. Student. He is a student of T.H. Morgan and he postulated gene maps or the chromosomal maps. What is this chromosomal map? Suppose the chromosome. On this chromosome, at one place A, B gene, C gene, D gene, they are present. So, chromosomal maps that are useful to distance between two different genes. Distance between A and D, A and C, A, B. It is possible by using these chromosomal maps. Now, these chromosomal maps that are widely used in Human Genome Project. So, linkage that maintain same genotype in all offspring generation to generation without any change. Phenotypic ratio. In a linked organisms, there is a 3:1. Diherbed cross, the linkage when appear, the phenotypic ratio 3:1 instead of 9:3 3:1. That means 3:1, that means they reflect the parental characters only.